place that up here yet. We will be talking about the eye. Now the eye is something normally I like to cover at the end. I just had to move it in order to make things balance out for exams and cover it a little bit earlier. Turns out I've got to cover it even earlier with the med students earlier than I would really like to be doing so. But the eye, as we take a look at it here, the eye. We take a look at it here. It basically you can think of as a tri-layered hollow ball. And we'll start off talking about each of those layers. There are some accessory structures or other structures that don't necessarily fit into that layered anatomy of it, and we'll describe those as well. But basically what we're doing with the eye is you're going to have light coming in here, coming in this direction, and you're going to want to focus that light. You're going to focus it a good bit here, a good bit here, and you want to focus it on the retina back here, which I always like to make an analogy of. That's the movie screen. That's where you will detect the light that is coming into the eye. Very interesting. We get to the development of the eye here towards the end of the semester as well. But the three layers. Now, layers, we've had different ways of defining them. Strata, tunicas, zonas. We're back to tunicas here for the eye. There are three tunics that make up the eye. And this is using the more anglicized term, the fibrous tunic or the tunica fibrosa. That's going to make up that's going to be made up of the cornea anteriorly and the sclera, the rest of the eye. You can see the blue layer is continuous, surround the outer layer surrounding the entire eye. And then there's the tunica vasculosa, as the name would imply. That's going to be a vascular layer. The bulk of it's going to be the choroid back here in the posterior portion of the eye. But you come forward, it's also going to include the ciliary body as well as the iris. Okay? And then the internalmost coat here is the tunica interna, which they're just labeling here as the retina. And they're talking about the fact there's a pigmented layer, neural layer, where the neural layer has three components to it. We'll describe all that in detail here a little bit later. Now, beyond the tunics, there's other structures we'll talk about. We'll talk about the lens, look at its histology here. We'll talk about the anterior and posterior chambers, which anterior chamber will be right here, posterior chamber will be right here. One of the confusing things, and I've had to correct clinicians coming in talking about the eye before, there's cavities. There's an anterior cavity here and a posterior cavity. The chambers are subdivisions of the anterior cavity. Okay, and we'll look at those, we'll define those a little bit better when we get here a little bit further. The posterior cavity, I like to refer to it as a vitreous cavity. They're labeled here vitreous humor. That's what's in it, and that keeps me from making the mistake of calling it the posterior chamber, which is going to be between the iris and the lens. This little area, empty area down in here, will be part of the posterior chamber. This back here in the red, with the yellow to it, is the posterior cavity that contains the vitreous body. And then there are accessory structures outside of the eye. There is the conjunctiva, which you see a little bit right here. Have an inflammation of that, this conjunctivitis. We'll talk about the eyelids. We'll also talk about the lacrimal glands. We'll look at those histologically. Let's go back and start talking about the histology, though. Look at the histology, though. Starting off with the tunica fibrosa. Remember, anteriorly that was the cornea, posteriorly the sclera. Well, this is the cornea. And the cornea here is your primary focusing media of the eye. It's the one that does the most bending of the light waves coming in, or let's use proper physics term, the most refraction of the light waves coming in. You think the lens is what really, does refraction? No, the lens does the fine tuning. The bulk of it is done here by the cornea. Okay, if we look at its layers here from superficial to deep, here's the outside. So it's lined by, you look at that, multiple layers of cells, looking near the surface there, flattened like pancakes, stratified squamous epithelium on the surface right there. And you look at it, higher power magnification over here, It's a non-keratinized stratified squamous. I mean, keratinized is what we found in the skin, and you'll find it other places if you have a lot of friction occurring as well. But it's what we find in the skin, so you expect this to be a non-keratinized stratified squamous here. It contains many nerve endings that actually make their way up the epithelium. That's why if you scratch your iris, it is scratch your cornea, it is very painful. And the flip side of that, though, is it's got a very high rate of cell division, so it repairs itself quickly. Okay? I've been told that you only do that one time. Take that weed eater out or whatever without wearing eye protection one time when you scratch the cornea because of how painful it is. All right. Now the just deep to it, if you look at the high power magnification, you can see at the lower power over here, there is a very thick basement membrane here, and that basement membrane is unique. We're going to see quite a few things that are unique within the eye, some things that break rules we've talked about previously. But this is an unusual basement membrane. It's got, actually has some ground substance in there, and it's got more reticular fibers than normal within that basement membrane. It stands out, even at low power magnification over here, you can see it as a negative image. You can see the negative image of it right here. It's called Bowman's membrane after the histologist at first described it. The bulk here of the cornea, if you look at the actual thickness, here's the epithelial thickness, here's that Bowman's membrane. The bulk of it here, if you looked at that and you had to describe it, you said it looks a lot like tendon, wouldn't you? Well, it shares a lot of qualities in tendon, but it's actually different as well. We refer to that layer as a stroma. It's responsible for the bulk of the thickness there, and you'd be correct in terms of the pink you're seeing in there are collagen fibers. And the nuclei you're seeing in there, the nuclei of the fibroblast in between the collagen fibers, and so they kind of line up into rows. Where it's different than tendon, all the collagen fibers were running the same direction. This may look like they're running the same direction, but you've got some running across horizontally here, and then you've got a layer just below it running out at you. And it's alternating layers running perpendicular to each other, okay? Kind of like plywood, okay? If you look at the grain in a piece of plywood, it's multiple layers, and they run the grain in alternate directions. It just gives it extra strength. And you think, this is providing the outer covering, going to be filled with pressurized fluid, okay? And it's just there to provide strength rather than having a dense irregular connective tissue to provide a capsule to provide strength. We want more of an ordered arrangement here. We've got light passing through it, and so we'd like to have a more orderly arrangement than a random arrangement of dense irregular connective tissue. But it's kind of serving the same function here to a great extent. All right, if we follow it on down, and then we come over here, this is the bottom portion, higher power magnification. If we look here, before we get to our inner epithelial lining, there is once again, a thick basement membrane here called Decimase membrane. It doesn't stand out to the same extent because it doesn't give us as much of a nice negative image as the Bowman's membrane, but it still is distinct enough you can see it. You can see it here, and then once you see it here, you can even make it out over here on low power magnification a little bit. It's got more elastic fibers than a basement membrane normally has within it, yet some thicker and a little bit different composition. We find plenty of things that are just a little bit different in the eye than other places in the body. And then the epithelium down here, which would be on the inside of the eye, lining that anterior chamber of the eye right here. We refer to that as our corneal endothelium. Endothelium, remember, is the simple squamous that lines blood vessels, okay? We use the same term here, endothelium. They're not related per se. They're not developmentally related or whatever, but it's a simple squamous, but a fat squamous, okay? It's not a short cuboidal. Simply, you look at the nuclei, there's still a single layer of them, flattened, long axis parallel to the surface. There's just more cytoplasm to them, so they're a fat squamous compared to a thin squamous like we often will see. All right, the next thing to talk about here, define at least, is the limbus. Okay, here is, come over to this side, image I guess first would be the best one to describe it. If we look down here, lower left-hand corner, here's the cornea that we just described. It comes back here and it is continuous then with the sclera. Okay, right there where it meets the sclera is called the limbus, and there's a lot of things going on at the limbus. We're going to come back to it multiple times. If you look, it's where the conjunctiva stops. The conjunctiva does not cover the cornea. We're going to see a canal of Schlemm in there. We're going to see that's where the iris begins. Just a lot of things going on at the limbus. The limbus is a junction where the cornea turns into the sclera. Okay? And we'll come back to that, like I say, quite a few times and describe it. The sclera was about 5'6", and don't memorize numbers. It's the bulk, though, of the tunica fibrosa here. Here's back further in the eye. Here's the inner hollow of the eye. Up here would be the vitreous cavity. So here is the sclera. Once again, you look at it there, it's a lot of collagen fibers, isn't it? Okay. We talk about things that are exceptions here in the eye. Why do we have this tough fibrous outer coating here? Well, first of all, you've got fluid in the eye. It's under pressure, and so you need a tough coating out there. But normally when we talk about muscle's got an origin in bone, an insertion bone, and contracts, it pulls two bones closer together. One of those exceptions would be your extraocular muscles that move your eyes. They originate on bone, they insert though into the sclera. Okay, and so if you think of muscles pulling, it's not pulling with a great deal of pressure, but it still needs something to be able to pull against. You need a tough fibrous layer there to attach that muscle to so it can pull against it without damaging it. All right, that moves us then into the tunica vasculosa. Another term for that is uvea, and we can see part of it right here. Remember that tunica vasculosa was the choroid ciliary body in the iris, okay? And so we'll start here with the choroid in terms of that. Well, choroid's in the posterior part of the eye, so here's the sclera. This layer right here, this darkly pigmented layer, is the choroid. We'll come back, we'll see pigment in many places throughout the eye, very dark pigment there, and that dark pigment is the same, whether you've got very dark brown eyes, whether you've got very pale blue eyes, that pigment is going to be exactly the same, all very dark here. And all of the open spaces you see within here are blood vessels. Tunica vasculosa, that makes sense. It's highly vascularized. Why is it highly vascularized? Well, here's a retina up here. We're going to see there's 1, 2, 3 layers of nuclei up here. Those are all neurons, and you know that neurons are major users of energy, right? That resting potential they have to maintain to generate an action potential is going to require a great deal of ATP. That means you need a lot of energy, means you need a lot of oxygen and nutrient supply, correct? That nutrient supply is right here within the choroid. Okay? That blood supply is right there within the choroid. That's why it's the tunica vasculosa. All right, and I was talking without looking at the slides. Once again, here would be the sclera. There's that choroid right there we just looked at. Anteriorly will be the ciliary body, and then the iris. And then use the same slide again as I talked about the choroid and the histology of it. All right, let's look then anteriorly at the ciliary body. Okay, here's that yellow sclera right here. The sclera would be the white of your eye anteriorly. But here would be the choroid coming forward, and then it enlarges. Now when it enlarges, it's often described when we're looking at it, in a section like this as being wedge-shaped. Realize it's a ring all the way around the eye though, okay? Yeah, it's got a wedge-shaped cross-section to it like this, but it's a ring going all the way around. It's coming out of the front of the screen here and coming back around over here as well. And so sometimes students get confused. He said it's wedge-shaped, but then it says ring-shaped here. But yeah, the cross-section, it is wedge-shaped, but in the entire structure of it, it's a ring going around the front here of the eye. All right? Now, when we talk about the ciliary body here, it's got two major components to it. There is the body itself, which contains ciliary muscle we'll come back to in it. and then it's got these little tiny projections, these little peninsulas coming off of it here. They're referred to as ciliary processes. Those ciliary processes, if you take a look at it, have little teeny tiny ligaments coming out here, little teeny tiny string-like structures that we're going to call ligaments of Zen that connect to the lens out here. In fact, the lens is suspended in place by all of these ligaments of Zen connecting it to the ciliary body. Connected to ciliary processes, which are connected to the ciliary body. All right, here's higher power magnification of what we've been talking about. Here's the ciliary body right here. Here's the ciliary processes coming off. Here's a little bit different magnification at the very front of it here. This is the very front of that ciliary body. Here would be the iris coming off of it here. Here's a limbus right here. And every now and then you do get just a little bit of a piece of a ligament of Zen in there. I think they're calling it a suspensory ligament, which is a newer term coming into play, but that ligament of Zen. I'll show you one in lab where they happen to catch it all the way from the ciliary process to the lens up here. Okay? But typically you just see little bits and pieces of it, just the nature of making sections right there. If we look at it in terms of higher power magnification, look at the ciliary processes, if you take a look, it's got a stratified cuboidal epithelium, 2 layers. The bottom layer, the deep layer, is highly pigmented. Once again, we'll talk about that pigment. That pigment's going to run continuous up here on the iris. It's also going to run posteriorly, be continuous with the pigmented layer of the retina. And then that superficial layer that's non-pigmented right there, those luminal cells, those are what make the aqueous humor. Aqueous humor is a water-based solution, kind of like selective filtration, once again, from capillaries within the ciliary processes here. But it is the fluid that fills the anterior cavity. In other words, the anterior and posterior chamber of the eye are filled with this aqueous humor. Okay, that aqueous humor is important as a filler. It's also important as a nutritional medium. Okay, there is not going to be any blood vessels passing through anything that light has to pass through. Okay, why? Well, think again of how we describe the eye. You're focusing light with the cornea and the lens onto the retina, the movie screen in the back. You know, rather than having TVs, if I had projector mounted from the ceiling and projecting the light up on the screens, what's going to happen if I've got blood vessels passing through, blood cells passing through that beam of light? It's going to create shadows, isn't it? Okay, and that's why we can't have that. We cannot have vascularization of structures. Light is passing through. What's light passing through? Cornea and lens. They're avascular. How do they get their nutrition? That's the job of the aqueous humor. Okay, aqueous humor is a filler and a nutritional medium. We'll come back to that concept of a nutritional medium here in just a little bit. All right, the next thing to talk about here would be the idea of the muscles down here within the ciliary body. Smooth muscle down here, making up what we call the ciliary muscles. And what they do, when this muscle contracts, it pulls on these ciliary processes, which are attached to the ligaments of Zen, which are attached to what? The lens, correct? And that is what will focus, that is what will focus the light properly on the retina. Okay, I couldn't 5% realize that this is not a high resolution drawing, obviously, but it showed what I wanted to show, and I'm not getting into a great deal of detail here. You know, normally if you've got, you know, a camera that is focusing, particularly an old camera, if I had the old overhead, you guys familiar with overhead projectors? Okay, yeah, I've got the acetate laying down on it, and I've got a lens up here, and it moves up and down to get the right focus here. Realize your eye doesn't change in and out and so on in terms of finding the proper focal length for it. Instead, what it does is changes the shape of the lens. And basically, that's what we're seeing at the bottom of this right here, is the idea that in order to accommodate for different light, we'll pull and stretch that lens out, or we'll relax the ciliary muscles and let that lens become more plump, okay? And that changes the focal length and allows us to accommodate between far vision and near vision in terms of focusing that light on the retina. And you guys would have handled this. I'm not going to get into the physics of it. If you remember the physics, whether it's something close, the rays of light are angling outward, where something far, they're coming in parallel. And that's the difference here in terms of focusing that light on the retina. Rather than having lenses that we've got, if I had a magnifying glass, I'm moving it up and down to find the proper focal length. Instead, what I do to find the proper focal length with the lens, I change the shape of it, okay? It's a little bit different here, and that's the job of those ciliary bodies. Pull on the ciliary processes that pull on the ligaments of Zen that then change the shape of the lens. Okay, anteriorly then, coming off the ciliary body, here's a ciliary body right here, coming off of it anteriorly will be the iris. The iris is what gives you the color of your eye, brown eyes, blue eyes, or whatever. And of course, the aperture in the middle that the light is going to be passing through to get to the lens, to then get to the retina, is the pupil. If we look at the iris histologically, here it is, or here it is even more so under high power. The iris is the boundary between the anterior cavity, our anterior chamber, I just used the word incorrectly, the anterior chamber and the posterior chamber, okay? So here in the anterior chamber, if we take a look at the iris basically controls, controls the amount of light going into the eye in terms of, is that pupil constricted? Very bright light, I'm getting too much light in the eye to see, let's control the amount that goes in. Or it can be dilated. For instance, I'm outside at night, I'm having trouble seeing because it's dark, and so pupil will dilate in order to allow more light to come in. Okay, that's the job here of the iris. We look at it histologically. What type of epithelium is lining the surface right here? Well, I can't tell low power, so my best guess would be simple squamous epithelium, right? We'd be wrong, okay? This is going to be one of the exceptions. There is no epithelial tissue lining that surface. There is a simple squamous during development. We lose it. I don't have an explanation in terms of why we lose it, why we're better off with this design. All I can tell you is here's a surface that has connective tissue on the surface, not an epithelial tissue. And you can see that, this higher power magnification, it does not have an epithelial lining at all on that anterior surface of the iris. All right, the bulk of it is going to be the stroma. Use that same term, stroma. Stroma is just defining a connective tissue layer. There's plenty of fibroblast in there within that stroma because it is connective tissue. It's going to be highly vascularized. It's also going to have melanocytes in here, and it's the melanocytes in here that produce the brown, the blue, the green eyes, the hazel eyes, every shade in between, okay? There's a pigmented epithelium on the back side here. This is going to be continuous from what we saw on the ciliary processes, except now both layers are pigmented. But you look at that and you can't tell me that's a stratified cuboidal epithelium because the pigment is so dense you can't see anything, right? All right, but this layer down here is not the layer that gives you the color of the eyes. Everybody has this very dark brown, almost flat pigment here. It's the melanocytes up in here that gives you your eye color. Okay, now the other thing we find in here, within it is we're going to find muscle, and actually two different sets of muscle that we find in there. There is the constrictor pupillae, sometimes called the sphincter pupillae in some sources, and there's the dilator pupillae. In other words, one's going to constrict the pupil, one's going to dilate it. And basically what you've got in terms of constricting, you've got it centered as a ring around the eye. And this one is actually myoepithelial rather than true smooth muscle. But as those muscle cells get shorter, it's going to make that opening smaller, isn't it? Okay? And so it is going to constrict. Here is normal. If you contract the sphincter here, it is going to make the pupil smaller. On the other hand, you've got smooth muscle. The dilator are arranged more like spokes on a bicycle tire. And so when those get shorter, when those contract get shorter, what are they going to do? They're going to pull and make the pupil larger, aren't they? Okay. This one is under parasympathetic control. You're resting and digesting. Let less light in the eyes allows you to rest better, correct? The dilator is under sympathetic control. While your fight or flight response, your pupils get dilated so you can let more light in to see better. Okay? And you can see these, back a slide. You can see that you don't have to distinguish between the two histologically, but you can see what looks like smooth muscle. Technically, this one is myoepithelial cells instead of smooth muscle, but it looks like smooth muscle. And right here and here. So you see different muscles in the eye, sets of what looks like smooth muscle in the eye. That's there to control the size of the pupil. All right. The next thing we need to talk about here at the limbus is the canal of Schlemm. All right, in order to understand why we need the canal of Schlemm, let's talk first about the circulation of the aqueous humor. We said we've got two things that light's passing through here, the cornea and the lens. They both have to be avascular, so we don't create shadows on our movie screen. Okay, and that aqueous humor acts as a filler here. Here's the iris, so in front of it is our anterior chamber. Back here is our posterior chamber. We see the yellow right here would be our vitreous body, or posterior vitreous cavity here. But we're making the aqueous humor here on those ciliary processes, and it moves up and around and eventually into the anterior chamber. If it's a nutritional medium, that means we've constantly got to be supplying either more glucose to it or replacing the medium, correct? We do the latter. Okay, we don't just put more glucose in there, we replace the medium. And so basically what's happening here is we're constantly making aqueous humor here on these ciliary processes. How do we get rid of it as we're constantly making more? That's the canal of Schlemm, which is right here at the limbus, is where it's located. We look at it histologically. I mean, you may have seen it drawn in elementary school drawings. I've seen it where it looks like a little tunnel, to drain that fluid out. It doesn't actually look like that. It's a vein. Embryonically, it's a vein that loses its arterial supply, but it's still connected to the venous side, and it doesn't actually come to the surface. Sorry, I've got this cold. A lot of you have it. It hangs on. Okay? You've got this aqueous humor here, and realize you're constantly producing it, so it's producing somewhat pressure. You can't have too much pressure. Okay? That's glaucoma. But basically, when it's under pressure right here, it percolates across that endothelial lining, across the stroma, gets into the vein / canal of Schlemm, and from there it can be returned to venous circulation. Okay? So it has to percolate across tissue. Mechanism, from my understanding, is not known in terms of how this really works and the details of it. It may be just enough pressure in here, it pushes some of that fluid diffuse across the tissues here. Once it gets into that vein / canal of Schlemm, it's got passageway to go back into circulation. All right, let's spend some time on the tunica interna. Tunica interna, or the retina. Here we're at the very back of the eye. Here's our sclera in white. I purposely used a lot of different drawings here just so we can see it from different perspectives. Right here would be my choroid. You can see all the blood vessels they've got in it. And then this right here would be my retina. Okay, this would then be an enlargement, a drawing of the retina. Here is my choroid. And the retina has a layer of pigmented cells right here. And then 1, 2, 3 layers of neurons. Okay, in terms of making up that tunica interna. All right, let's take a look at it from the drawing that your textbook uses. All right, I love it when they do this. I mean, this is something fairly complicated. And so what they did was they took a micrograph here and did the drawing exactly with it. Okay, where things line up across. Here would be the choroid down here. If you look, see a little bit of what looked like the choroid before, correct? Here is my pigmented epithelium, or my pigmented cells. You can see them right here. And then I've got 1, 2, 3 layers of nuclei. 1, 2, 3 layers of nuclei. Okay? Now, histologists are anatomists, and as anatomists, they want to name every single layer and line they see in there. You're not going to have to know all the names of those per se. I want you to know what functionally will be in here. We'll go through that as we go through the various slides here. Okay, we're going to start off down here with that pigmented epithelium. Okay, higher power magnification of the image before. Here's our pigmented epithelium right here. All right, that pigmented epithelium is going to be simple cuboidal epithelium. And once again, the pigment in there is melanin, okay, a very dark form of melanin. And we've seen that dark pigmentation all over the place, haven't we? We've seen it on the posterior surface of the iris. We've seen it on the basal cells of the ciliary processes. We've seen it in the choroid. We see it here. Now's the time we're going to say, what is it here for? Okay? Let's talk a little bit about how the eye works. Basically, you've got light coming in. Let me show it in the drawing up here. Light coming in, and it turns out we're not going to detect the light coming in until we get down here. Okay? Over here, until we get right to here. Now, when light comes in, got many things that has to pass through before it gets to our receptors down here. But it could potentially miss a receptor, and what we don't want to have it do, light come down and hit down here and bounce back off, and then detect it on the rebound. Okay? If we see rebound light, in proper term, reflected light, you know, if we're out, we get that a good bit on water, we call it glare. That's why if you're out in the water, you typically have polarized sunglasses that will take out that reflected light so that you don't see it. Takes care of the glare. We'll come in the eye, we only want to see the light as it is coming into the eye. We don't want to see it after it's bounced around off the wall of the eye somewhere, do we? And so how do we prevent it from bouncing off the wall of the eye? We make the walls very, very dark with melanin. Kind of like that black top. When sunlight hits that black top, the light doesn't bounce off of it. The black top collects all that light. That's why it's black. Because the light, no light's bouncing back. You learn that in physics. You also learn that's why it heats up, correct? Because it stores all that energy in the light. But we're not worried about heating up here, but we want to store, we want to capture any light that isn't detected by our photodetectors, our light detectors. We don't want it bouncing around off the walls of the eye, so what do? We make all the walls very dark with all this very dark melanin. Okay? Makes sense, doesn't it? Okay? And so that's the job of the melanin. That's one of the jobs here of this pigmented epithelium. If you look at these cells, over the drawing here, they've got these long microvilli. Some books will call them stereocilia. They're not really as long as true stereocilia, but these long microvilli here, you get a little bit of impression here that that's where you actually have the pigment within those microvilli. I'll come back to talk about the function of those in just a little bit here. There's also a thick basement membrane just deep to it here called the Bruch's membrane. Bruch's membrane is a basement membrane just deep to that pigmented epithelium. Okay. Here's just a little bit higher power, those pigmented cells, once again. You look there, cuboidal epithelium, got a kind of a rounded nucleus, and you can get the idea of those long microvilli in here, and it's in those microvilli that you have all of that melanin. All right, let's move on then and talk about the neural component here of the retina. Here's my sclera up top, here's my choroid, here is my pigmented epithelium, and then I've got, once again, 3 layers of neurons. You see them by layers of nuclei, 1 layer here, 2nd layer here, a 3rd layer right here. You're gonna have to know the names of these. These are my photoreceptor cells, these are my rods and cones, these are my bipolar cells primarily, and I'll name all these again, and these are my ganglion cells down here. Okay, 3 different layers of neurons there. If I want to talk in a little bit more detail about the photoreceptors, if you take a look at them here within this drawing, you can see there are very thin pencil-shaped ones. Those are called rods. And then there are thicker ones here that are called cones. And if we look over here in the micrograph, cones are the large pink things you see in here. The little tiny pink lines you see coming down would be the rods. All right, here they are in a little bit more detail here. Here is the rod coming down. Here is the cone. They're both surrounded by a particular cell right here, a supporting cell called a Muller cell. And you can see the cytoplasm over here. They cut it off here because the nucleus is way up off the top of the screen. Okay, it transcends, its nucleus is going to be up where the bipolar cells are. But a supportive cell there called a Muller cell. When we look here at the rods and the cones, they have an inner segment and an outer segment. And that inner segment here, they would be right here. Both of them are on the outside of the nucleus. Here's the nucleus of rods and cones here. And so this would be the outer segment, and then here are my inner segments. Okay, I did that backwards. Here's my inner segment, here's my outer segment down here. The inner segment is where we've got all the cell machinery, the mitochondria, the smoothie or the rough ER, the Golgi, and all that. And the outer segment right here, it's made up of little vesicles that are flattened and stacked on top of each other. All right, they're made up here with the rough ER, the Golgi, and so on. And then they're stacked on top of each other, and as one is made, it's stacked here. And so the newest ones would be up here. The oldest ones would be down here in the tip of each. What is in those little vesicles there? There are photoreceptive pigments. There are pigments there that detect light, and when they detect light, they change their structure, and that is how you detect light. Now the rods is a little bit different in terms of the pigments there. Rods have pigments that can detect very low levels of light, but you only see from them in black and white. Hey, think about it. If it's dusk or at night and you see your roommate across the parking lot, you might be able to tell your roommate's wearing a dark shirt, but you can't tell if it's that dark red or that dark blue shirt, right? In low light, you use your rods. They don't need as much light, and therefore they only function in black and white. Whereas the cones, they see in color, but they require a lot more light in order to function. Now, the outer segment here of both the cones and the rods are going to be cradled here by the microvilli. Now, there's a little bit of an artifact here we've had a little bit of separation between the pigmented epithelium and the outer segments of the rods and cones. Okay, that's quite common as you're making the slide. If you ever heard of a detached retina, that's where it detaches, right in that layer right there. Okay, that's where it's least held. It's held together least firmly there compared to other places of the retina. And so, yeah, detach a little bit right there. That's important because you've got the outer segment here of a cone being cradled by these microvilli of the pigmented cells, and a second job of those pigmented cells, second job, we've already said they absorb photons of light that make it pass, as do other pigmented layers here within the eye. But they're responsible for removing the stacked vesicles down here at the tip. Remember, these are the newest ones up here, correct? These would be the oldest ones down here. Well, that photoreactive pigment, as it's used over and over and over and over and over again, reacting with photons of light, eventually gets damaged, gets worn out, and so we remove the older senescent vesicles down at the bottom here. That's the job here of this pigmented epithelium. It phagocytizes the tips of these in order to remove the old, worn out photoreceptive pigments. Almost neat being an engineer on this, isn't it? I mean, just amazing how everything works here. And yet, it all makes sense, doesn't it? Okay, now I'm not so much going to go in terms of describing the physiology of how this works. I mean, Dr. Olgan has done that with you. Remember, it's not so much that you generate action potentials here as much as you are always generating action potentials. You generate less when light comes in. I'm not going into that in any detail. But basically, once we detected light in here, how do we get that message to the brain? Well, it's first detected here in the rods and the cones. Then it has to go through the middle layer here, which are bipolar neurons. Now, those bipolar neurons are simply interneurons. Interneurons connect two different neurons, right? They're going to connect the photoreceptors, rods and cones, which are neurons, to the ganglion cells out here. Now, we also are going to have some other cells in here. This one shows us horizontal cells and amacrine cells. I'm not going to go into this in any type of detail. I'm just going to say those nuclei are in there. The only thing I'll say is these help you see nice, crisp, clear lines. You've seen images or whatever where they have little dots that kind of fade from the, let's say, the letters into the t-shirt or whatever, where it's no longer a crisp, clear line purposely. You'd see a lot of that in terms of non-crisp, clear lines when you look at things without the amacrine and the horizontal cells. The Muller cells we talked about earlier are also, their nuclei are also within that layer right there. But the most of the nuclei you see in there are going to be the bipolar cells. Okay, and so there they are right here. Primarily bipolar cells, but as well as some amacrine cells, horizontal cells, as well as some Muller cell nuclei in there. And then the next layer out nuclei here, and this would be the closest to the vitreous cavity of the eye. Those are going to be the ganglion cells. And those ganglion cells, if you take a look at them, after they receive the message from the bipolar cells, their axons are then going to carry it along the way on the surface. So this layer up here would be the axons of the ganglion cells. They're going to carry it along on the surface until it leaves the eye and the optic nerve. And we'll look at that in just a little bit histologically. Okay? Now, if you were going to design how the eye would work, I've got a bunch of cool design features, but if you were going to put the photoreceptors, where would you put them? Would you want to put them down here, where light's got to pass through everything to get to them, or would you want to put the tips of the rods and cones up here? We'd probably want to put them up on the surface, wouldn't we? I mean, we're going to lose a lot of light passing through here. Well, that's the way our eye is designed. We'll see embryonically how it comes to be that way. But it turns out we've got one portion of the eye here, one portion of the retina, maybe I say, called the fovea centralis. You know, this was the image that we looked at before. Light coming in, and then the message is represented by the blue arrow going out that we detected light down here. And we said light has to come through the layers of nuclei here of the ganglion cells, layers of the bipolar cells, layer of nuclei here of the rods and the cones before it can get detected down here within the photoreceptor layer, correct? And so the fovea centralis is a little bit different. If we look at it here, here's my fovea centralis. If I look, if I look right here, the pointer is going through right here. That would be my pigmented epithelium. And so just above my pigmented epithelium right here, here's the layer that's got the inner and outer segments of the rods and cones where the light's detected. This would be the layer of nuclei of the rods and cones. I look right here, I don't really see a whole lot of the bipolar cell nuclei I do on either side, don't I? And I don't even make out a third layer up here of the ganglion cells. What's happening at the fovea centralis? Rather than everything being directed by a type A person going at right angles to everything here, instead what we've got here is these cells, rather than being oriented straight up and down, are oriented diagonally. Okay? And so as they're oriented diagonally, the light coming in, striking right here of the retina and the fovea centralis, doesn't have to pass through all those layers of nuclei anymore, does it? It still has to pass through the layers of the rods and cones, but the bipolar cells and the ganglion cells, their nuclei over here on the side, the light coming in right here at the fovea centralis, doesn't have to pass through those layer of nuclei, does it? The fovea centralis, when you're focusing your vision on something, you're focusing that light directly on the fovea centralis. This is where you have the greatest visual acuity. It's where you see the most detail, because the light didn't have to pass through all the different layers of nuclei. Okay? For instance, if I am focusing on the clock in the back here, I've got peripheral vision out here. I mean, I can see I've got my hand up here, I can see I've got my fingers moving, but that's not on the fovea centralis. That part's not being focused on the fovea centralis, the clock is in the back, that's where I'm focused. I can see my hand out here, but I couldn't tell you, if you came and manipulated my hand, I couldn't tell you if I had 1, 2, 3, how many fingers I've got put up on my peripheral vision. Okay? Why? Peripheral vision has to go through all the layers on the right side in that drawing there, whereas what I'm focused on have a lot more visual acuity because it doesn't have to go through all those layers. Does that make sense? Okay, when you get to the clinical lab where you're looking with an ophthalmoscope at the retina, you can actually see the fovea centralis as kind of a small yellowish disc. It's called the macula lutea there. Macula lutea is just describing what you can see with a ophthalmoscope of the fovea centralis. Okay, now that fovea centralis, if we look at it, it looks like a little bit of a, from the surface here, it looks like a little bit of a divot into the surface of the retina, doesn't it? It doesn't have all the layers of nuclei there. There's something else that has a divot within the retina, and that is what we call your optic disc. Okay, let's orient ourselves again. All right, here's my scleras, the U is the choroid or uvea. And so here's one, here would be my retina. You can see I can make out a layer there of the pigmented epithelium, but right here, this would be my rods and cones, my bipolar cell nuclei, and up here would be my ganglia cell nuclei. Okay? Now if I look, the retina, it's got a little bit of a divot in it, just like we kind of saw with the fovea centralis, correct? But if we take a look, if this is the rods and cones, this is the rods and cones nuclei right here, and you can even see a little bit of the rod and cone portions hanging out below, can't you, in that little pink and white area there. Photoreceptors don't extend all the way across here, do they? Okay, what did we say was on the surface up here of the retina? This layer up here consisted of the axons of ganglion cells. Those axons of ganglion cells at the optic disc are coming here, and they're turning backwards into the brain to become the optic nerve. Okay, how do you distinguish this from the fovea centralis? There's no rods and cones coming across here. Okay? Fovea centralis, you still had, within that middle of that divot, you still had nuclei of rods and cones, correct? You had to, because that's where you're detecting the light. Here, this is sometimes referred to as your anatomical blind spot, which is covered up because you got two eyes. Anatomical blind spot's not in the same place. You might say, wait a minute, I closed one eye, I don't have a blind spot. Go to YouTube and you can find something there to illustrate, yes, you do have an anatomical blind spot, you just fill it in with what you think is there, okay? Remember my second grade teacher, so tall, literally, Sister Miriam Joseph. She had all kinds of neat science things, and one of the things she had was showing us our anatomical blind spot, okay? And so, you know, it's there, like, once again, and why is it there? If you focus light right here, any light focused right here, can't be detected because there's no rods and cones, correct? If we take a look at these axons coming back here, all of a sudden, there's a line right across here where it gets a little bit of a darker pink. That's where we're gonna myelinate those axons. Why don't we myelinate them up here? Well, light has to pass through them in order to be detected by rods and cones, correct? Why add another layer of nuclei in there, myelinating Schwann cells, really be oligodendrocytes here because it's still part of the central nervous system. Why add another layer of oligodendrocyte nuclei? We won't do that until we get down here, and then we'll myelinate those axons as they go back to the brain. Optic nerve is considered not a true nerve, but an outgrowth of the brain. That's why they're showing covering the dura mater, pia mater, and arachnoid mater right here. That's why I said the oligodendrocytes, not Schwann cells. We'll often see it say Schwann cells here. It's oligodendrocytes. That's an outgrowth of the brain. And then look to see what else. The only thing I haven't talked about is the fact that blood vessels will enter and leave the eye here. We're going to see during development. One will come all the way up and connect to the lens. We're going to lose that one. And also come over here and go to the uvea through the optic nerve right here. All right, let's talk about, whoops, I've talked enough. Before we take a break, let's talk